So here we go, topics 4 and 5 of Unit 2, the plasma membrane and membrane permeability. Just as a reminder, take a few moments here to review the macromolecule from Unit 1, the phospholipid. And to the right-hand side, remember that the phospholipid identifies really 4 major structures. First, that phosphate group, that PO4 group, and glycerol. Those two molecules together make up that hydrophilic head, that water-loving head, and then the saturated and unsaturated fatty acid tails that are attached to that phospholipid head that are hydrophobic. And those are just carbon-hydrogen chains. Remember, the unsaturated ones, the unsaturated guys right here, they're going to have more double bonds, where the saturated ones, think saturated with hydrogen. Remember, carbon, 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 and then off of each carbon come those hydrogens. So think saturated with hydrogens. And then if I remove one of these and I create this double bond, this carbon-carbon double bond, I'm getting more of this unsaturated fatty acid. To better help to see if we can remember things from Unit 1, the plasma membrane, answer a couple of review questions 2 through 4. All right, so the cell membrane, the plasma membrane, phospholipid bilayer, if you will. That was lightning. All right, it separates the cell's internal environment from its external environment. It's made up primarily of phospholipids. There's a lot of stuff in there, but it's mainly phospholipids. And phospholipids are called amphipathic. They're molecules that have the best of both worlds. They have that hydrophobic area and that hydrophilic area. So they're able to interact with water partly and the other part not. And it forms this bilayer, hence 2 layers. In science, we use a lot of models. So this fluid mosaic model is what is used to describe how the cell membrane functions as a structure. It's very, very fluid, which means it's held together by these weak, hydrophobic interactions. And therefore, it can move and shift. It's almost like if you were to walk across the waterbed. Lots of things can affect the fluid mosaic model, like temperature. Temperature will definitely affect its fluidity. And then these unsaturated hydrocarbon tails will definitely help maintain fluidity at low temperatures, because these unsaturated ones, they're very kinky. And when they're kinky, they can't form a tightly packed area. The kinkiness allows them to stay more separated from each other. I like to use the term like lasagna noodles. You know, lasagna noodles, if you just lay them out, they will stack on top of each other. But if you were going to stack little bow tie pasta noodles, they wouldn't stack very well because they've got a lot more kinks and shapes to them. Similar here. Buried inside of the fluid mosaic model is cholesterol. Now, cholesterol helps maintain the fluidity at high and low temperatures. It's very useful because at a high temperature, it's going to reduce movement, kind of create a more rigid structure by putting something in there that wouldn't allow the fluid model to move as much. But at a low temperature, it's going to reduce the tight packing of the phospholipids. So that all comes to one point, which what does mosaic mean? Mosaic is comprised of many macromolecules. So you may have family members that say the mosaic window. And on like the sides of churches, you can see windows that are comprised of all these little tiny pieces of glass that have a variety of colors. That's a mosaic. Or paintings where we've crushed up something and made a painting out of these little tiny pieces. That's what mosaic means, made up of many others. And that's what the fluid mosaic model is. It's got a lot of pieces in order to function in its structure. Buried inside of the plasma membrane are two different types of proteins. Well, there's two major types of proteins. There are integral proteins and then peripheral proteins. When they're integrated, those proteins are embedded into the lipid bilayer, which we can see right here. This is an integral protein. They too can have amphipathic properties, meaning that they can interact with this hydrophobic and hydrophilic aspect of it. Peripheral proteins are proteins that are not necessarily always embedded into the bilayer, but they're loosely bonded to the surface. And these guys right here, integral proteins, play an integral part in how the cell membrane functions. Not only proteins can be embedded, but so can carbohydrates. When cells communicate to each other, there's cell-to-cell recognition, and it happens in several ways, but one of them is a glycoprotein. That is a protein that is attached to one of the protein channels inside of the cell membrane. These are pretty common, they're very abundant, but on the outside of that integrated protein is a carbohydrate. It kind of works as a communication tower, if you will. It can also be in the form of a lipid. So when it's a carbohydrate that's bonded to the lipid portion of that phospholipid head, we call it a glycolipid. Those are not nearly as common. It's more common to have the proteins have these carbohydrates just like hanging out, wanting to interact with other cells near it. Plant cells can have their membrane definitely works different. It has some special features. The most obvious, and the one often students know, is that plants have cell walls. That cell wall covers its plasma membrane, and its extracellular structures are different. They're not found in animal cells. This cell wall provides a lot of structure and shape. It also protects the internal delicate components of plant cells, and it is responsible, if not mostly responsible, for regulating water in and out of plant cells. But its most predominant feature is that it will regulate the intake of water, and that it of course provides this structural shape. The cell wall is comprised of cellulose, and it's significantly thicker than the plasma membrane itself. It contains the plasmatisma, which is these little hole-like structures in the cell wall. They're filled with cytosol, and they connect adjacent cells.